So today we'll be talking about evolution. My name is Amias. I'll be going through this webinar today. So before we start, let's go through the definition of evolution. So the definition of evolution is descent with modification from a common ancestor. So we first need to talk about the different terms used inside this definition, and they are descent, modification, and common ancestor. So we talk about descent. Basically, descent just means that you pass on heritable genetic changes from one generation to the next generation. So this would be in terms of our alleles, either favorable or unfavorable alleles. In the word descent, it doesn't matter. It just means the passing down of alleles. Okay, genetic changes there refer to your modification, your next key term. So modification refers to the changes in your allele frequency within one population over time. And the keywords here are change in allele frequency and over time. So the difference in allele frequency, what leads to the difference, we'll talk about that later. And next term will be common ancestor. So the common ancestor refers to one common prehistoric ancestor that the different populations or the different species have in common. Okay, so what is descent with modification? So characteristics present in an ancestral organism, something that is not present now, that has died off already, ancestral organism, developed into different forms as a result of natural selection. And the process of natural selection we'll go through later as well. And how these different forms come about is because they face different environmental conditions, so different selection pressures. Okay, an example of it would be your 5-digit pentadactyl limb. So we see the pentadactyl limb in a whole range of species, from birds to frogs to humans, and of course horses and whales as well. Over all these different species, how can we still have the 5-digit pentadactyl limb, right? That's quite interesting. So we'll talk about that as well later. So evolution can be split into two very big ideas, which is microevolution and macroevolution. So people sometimes struggle with the difference between these two. The big difference between these two is just time. The only difference between these two is time. Okay? When evolution occurs on a smaller time scale, that means just a few generations, microevolution occurs. And the result of microevolution is no new species are formed, but there is a change in allele frequency. Just a minute change, not something that's too big, just something that is very, very minute, very small, very barely noticeable change in allele frequency. That is microevolution. Macroevolution, on the other hand, is something that occurs over a longer time scale, something that is thousands and millions of generations long. And the result of it is that new species are formed, and an example of that would be birds and mammals diverging from reptiles. Okay, next. So how then are we supposed to approach questions regarding how certain factor given in the question leads to macroevolution or speciation, right? So these are just some general steps, not the exact answer or the exact model answer. I'll just be going through the general steps, then I'll show you the proper answer. Okay, so the general steps, firstly, we talk about common ancestor, because we go back to the definition of evolution. We need the descent with modification from a common ancestor. Always talk about your common ancestor, your ancestral population. Next, talk about which factor led to disruption of gene flow. This is usually given in the question, so it's very self-explanatory. And you don't have to think much about it. You just copy from the question what they wrote. And the next idea you bring in is you have to talk about variation. If you don't talk about variation, the other parts of your answer will not make sense. So you have to talk about variation. So what does variation mean for a population? It means that some have a selective advantage. Those with a selective advantage are able to survive. And if they're able to survive, they're able to reproduce. If they're able to reproduce, they're able to pass on favorable alleles. And when they're able to pass on these favorable alleles, there'll be changes in the allele frequency within your population. And then your two subpopulations will evolve independently of each other, because if we go back to step 2, the factor that led to the disruption of gene flow, because now there's disruption of gene flow, that means one gene pool in one subpopulation and the other gene pool in the other subpopulation cannot flow and cannot mix together. Therefore, these two subpopulations will independently evolve. Okay, then we talk about the accumulation of different mutations. Because they are evolving independently, they accumulate different mutations because they are exposed to different environments and different selection pressures. Then over hundreds of thousands of generations, your usual phrase, your two subpopulations become reproductively isolated. They can no longer interbreed, and they can no longer produce viable fertile offspring. Therefore, they become two separate species. Because if we remember the biological definition of a species, it is when they cannot interbreed to produce viable fertile offspring, right? So this is the almost ideal answer, a model answer if you want to refer to it as a model answer. So the the factor here in the question would be either a physical barrier or reproductive barrier. So these would result in different types of speciation, your allopatric and sympatric speciation. So you have your allopatric speciation is geographical isolation. Sympatric speciation is reproductive isolation or physiological isolation. Okay, this is something that people struggle with differentiating allopatric and sympatric speciation. But it's basically physical barriers, reproductive barriers, respectively. So macroevolution, the next part of the answer. So this results in two different subpopulations in different islands. So if they're in two different islands, they're exposed to two different environments. Two different environments means there are two different niches for species to fill, and therefore they are exposed to different selection pressures. And that results in them not being able to interbreed because of the physical barrier. They're not able to interbreed, and they're exposed to the different selection pressures. Gene flow is disrupted. Okay, so this is definition of gene flow, right? So it reduces the differences in allele frequency between your populations, right? That is when gene flow works. But if gene flow does not work, which means gene flow is disrupted, your alleles are not able to transfer from one population to the other. Okay, so the difference in allele frequency increases. Next, variation. This is very important to put in your answer. People always leave this out in their answer, but it is very, very important to have inside. So, variation in your population means that there are some that will have favorable traits. These that have favorable traits or favorable phenotypes, they have a selective advantage and they'll be selected for. These advantageous traits will be passed down from parent to offspring. This is another part that people always forget to write, that is passed down. Because if I have a favorable trait, but my child does not receive it from me, then there won't be a change in frequency of favorable alleles. Okay, so that's why you always have to say that, yes, number one, there's variation. Number two, it is passed down. Your favorable traits are passed down. These two are where people always forget and always lose marks for. So, as the two subpopulations evolve independently of each other, their allele frequencies change, and they are subjected to genetic drift and natural selection, and therefore accumulate different genetic mutations. And then we go back to the phrase, hundreds and thousands of successive generations, the species become reproductively isolated, no longer interbreed to produce fertile, viable offspring. So the last part is always very crucial as well, because you're linking back to the definition of what a species is. Okay, now we talk about microevolution. So microevolution, again, is just the changes in allele frequency. So let's say a random mutation results in new alleles that, because of genetic variation, right? That led to genetic variation and difference in, for example, the size of wing, right? The one that is in arrows. let's say it leads to a difference in size of wings. So the presence of X acts as a selection pressure, and the X could be the presence of predators, a change in weather conditions, or, for example, the most an example would be the industrial evolution with your white and black peppered moths, if you all have that in your lecture notes. So individuals with Y have a selective advantage and are selected for. So it's the same, if you look at it, it's the same as macroevolution, just that now it doesn't result in a new species, right? It just leads to a change in allele frequency. So they're more likely to survive, reproduce, pass on their alleles, and over many generations, the frequency of Y alleles increases. The proportion of population with Y alleles increases, right? It doesn't mean that two separate species form because this is microevolution, only allele frequency changes. It's almost similar in the answering technique. It's just that at the end of the day, you're talking about the proportion of a certain allele or the frequency of a certain allele in the population. Okay, next we'll talk about convergent evolution. This was actually brought up in the bio chat about maybe 2-3 weeks ago. Someone was asking about convergent evolution. So convergent evolution basically is two different or more than two different species that are not closely related but look similar. So people always wonder why. You know, why does a fish look like a dolphin? Like they both have fins, but they're not closely related because they evolve independently of each other, right? They have analogous structures, not homologous structures. They have analogous structures because they live in similar environments and have adapted to the same way of life, right? That means they are exposed to the same selection pressures. Like your fish and your dolphin, they're both in the water and they both need a streamlined body to go through the water properly, right? So therefore they have similar shapes. Therefore they have the fins in place, right? It's not because they evolved from a very recent common ancestor, but it's because they live in similar environments. So this is convergent evolution. Of course, there's divergent evolution as well, but that one is the easier one, so we left it out of this webinar. But if you have questions on divergent evolution, you can always bring it up. And at any point in time, if you have any questions, you can just unmute yourself and just ask a question, okay? Okay. So, when we sent out the Google Forms, a lot of y'all asked about how we use molecular phylogeny how we use molecular evidence to evidence phylogeny, right? To prove that, sorry, let me pull up the, okay, how do we answer questions such as explain how molecular homologies and data allows for reconstruction of phylogeny? Okay, so this is the answer to that. So when we talk about different species and we want to see how closely related they are, we have to use a homologous gene, right? So a gene that's found in all your species that you are testing or that you are figuring the relation between. So let's say you want to see how similar fish and human are. Okay, first thing you need to do is find a homologous gene. So a gene that humans have, a gene that fish have. Okay, usually this gene has a high mutation rate, right? This is if we're comparing like very closely related species like humans and chimpanzees. Okay, so we look for a homologous gene, and usually, not all the time, usually they have a high mutation rate and are able to show significant differences between closely related species, closely related populations. And its mutations usually take place with clockwork regularity. And because it happens with clockwork regularity, let's say this gene mutates every 60 years, for example, then you see how many differences there are between the human and the fish gene, and then you can calculate, okay, maybe there are 1 million. So you take 1 million times 60 to determine the time of speciation, something like that. Okay, it's just an example, right? So an example of homologous gene between different species is your cytochrome gene or your mitochondrial DNA. So the difference between your cytochrome B gene and your mitochondrial DNA is that mitochondrial DNA, right, they do not undergo genetic recombination like your DNA. And the only differences in your mitochondrial DNA of a human and mitochondrial DNA of another species is solely due to the accumulations of mutations over time and at a regular rate, because mitochondrial DNA mutates with clockwork regularity. Other than your cytochrome gene and your mitochondrial DNA, you can also use your homologous proteins as well to determine the degree of relatedness. And after you determine the degree of relatedness, can you reconstruct phylogeny? Okay, so we also use amino acid sequences or DNA sequences, right? Because they are highly conserved. And then the question now is, if any one of you can answer, why we use DNA sequence, or why we prefer using DNA sequence instead of amino acid sequences. Can any one of you answer that? Just try. You can type it in the chat if you're too shy to talk. Does anyone want to say? Does anyone want to try? Is there a chat? Anyone want to try? Just try. Unmute yourself and try answering why amino acid sequences are not as preferred as using DNA sequences to determine the degree of relatedness. No one wants to try? Does Asher want to try? Asher, do you want to try answering this question? No one wants to try? Okay. Really? Nobody? Okay, fine. Okay, so why is your amino acid sequence not as preferred as your DNA sequence? Because, okay, let's think about it. You have your DNA sequence, and if we have a mutation in your DNA sequence, that will lead to a change in your mRNA sequence after it gets transcribed. And after the change in your mRNA, it may or may not result in a different amino acid being coded for in your polypeptide chain. Why is that so? Because your triplet codes are degenerate, right? You have different triplet codes that code for the same amino acid. So when there is a mutation in your DNA sequence, it will reflect in your DNA sequence, right? Because it is mutated along itself. So it will be observed on itself, on the DNA itself. But if we're talking about your DNA sequence having a mutation and how that would lead to a change in your amino acid sequence, that is further, right? That's more than one step further. That's about 2 steps further down. You have to first transcribe it. After you transcribe, you have to translate it, right? And because during translation, triplet codes are degenerate, a mutation in your DNA sequence may not lead to a change in amino acid sequence. Okay, so that's why we prefer using DNA sequences over amino acid sequences when we determine the degree of relatedness. So this is a question that likes to be tested because it is not as straightforward as people think. They might be stumped because they look at DNA and they look at amino acid and they're like, where's the link? Right? You think about it, it is because transcription and translation. Then that is your link afterwards. Okay. So of course, a caveat is that there has to be enough time for sufficient variation to have occurred to be able to distinguish between your different samples. Okay. And there's another question that they like to ask in the exams is, benefits of using molecular data. So I rank them in ones that make the most sense to me and ones that are more logical and easier to answer, easier to get me my marks, I feel. Okay, so you guys can look at all the points that I've put up and then see if you would want to rank them this way too. Okay, so why do we use molecular data? Because DNA sequences are objective, right? So your molecular character states, your ACGT, are unambiguous, right? They are easily recognizable and cannot be confused. Okay, so that is the first benefit. Second benefit is DNA sequence data is quantitative. That means that it can be easily converted to numerical form. So once you have it in numerical form, you can then use math and statistical analysis. So most of y'all would have had your lesson on statistics already. So yes, you can use it to statistically analyze your DNA sequence data, and then you compute it, and then you can use that to determine your degree of relatedness. Of course, it's inferred and it's not definitive. Okay, because you cannot how do you say this? You cannot directly say, oh, it happened exactly 601 million years ago. You can only roughly say, okay, maybe about 600 million years ago. Okay, so we will always advise not to say it can be determined, but we always advise to say it can be inferred. If your school does not make such a distinction, it is okay. Stick with what your school teaches you. Okay. Next is your next is that your DNA sequence data can be used to compare species that are morphologically indistinguishable. So that means that these two species look very, very similar, okay, because of convergent evolution, or because they are closely related. More often than not, it's because they are closely related. So if they are closely related, they may not be very morphologically distinguishable. Therefore, you can use your DNA sequence to compare between these two to tell you how related they are and to tell you which species they exactly are, if they are that morphologically indistinguishable. Okay, and the next one is because your changes in nucleotide sequences accumulate over time with clockwork regularity. Therefore, we can estimate the time of speciation. Again, we use the word estimate, not determine, because we were advised to. So we can estimate the time of speciation and place the speciation events on a time scale or in your phylogenetic table or diagram. So remotely related organisms can also be compared because they share common proteins like your cytochrome C, and also because all known life is based on nucleic acids. Okay. Next, we talk about biogeography. So to answer biogeography questions, you need to do three things. You need to, number one, account for the geographical distribution of your organisms, right? Link it to Darwin's theory of evolution. Next, you have to talk about descent with modification. Third, you talk about common ancestors. Okay, so again now you can see that it is. Okay, again you can see that it is. What's this called? You can see that it's the same thing as before, right? We talk about descent with modification. We talk about common ancestors. Just that now we're talking about biogeography. We're adding another aspect to our answers. Okay. So this is a very common question or a very common site in the world that they like to test, like the Isthmus of Panama. So yes, so two different species, AV and AT, are found in different places, and they are separated by your Isthmus of Panama, right? So why is it so that two different species, of course, there will be pictures and they will look very similar. They're fish, I believe. And so they ask you, how come there are two different species that have been formed, right? So you talk about the common ancestor first, or the ancestral population first. So there existed a common ancestor of both of these before they were separated by the Isthmus of Panama. Then, after the Isthmus of Panama formed due to continental drift or lowering sea levels, the common ancestral species got separated into two subpopulations. So once you explain that, okay, these two subpopulations came from one common ancestral species or one common ancestor, then you talk about your usual steps of speciation, okay, which we listed up above. I will go through that again, right? So they are divided into two different subpopulations because they are now geographically isolated. And because they're geographically isolated, there is disruption of gene flow. Okay, and because there's been disruption of gene flow, they're exposed to different environments, different selection pressures. Those with favorable alleles are selected for, right? So they evolve independently of each other because they're exposed to different selection pressures. And then you can add this in, okay, in your answers. You can add in the salinity, temperature, and nutrient levels. It can be given in the question, or you could just list it out. If this is an essay question, if you list this out, you'll be rewarded for it, okay? Or when they look at it, they'll see, okay, this guy knows what he's doing, this student knows what they're doing. You know, they're talking about the different environments. They're not just listing off the top of their head. So if you miss out another part of your explanation, or if another part of your explanation is not as coherent, they look at this, okay, they listed out salinity, they listed out temperature, nutrient levels. Because of that, you know what, I'll overlook this small little mistake they made, or this gap in explanation they made, right? So you can do it. It's not advisable if you don't usually finish your papers on time. If you do, you can write this down and it will definitely help you, okay? And they form two separate species afterwards. This is a very short answer. Of course, if you want to go through the entire process of speciation, you can, right? It just depends on the number of marks you have. But because this slide here is focusing on how to answer biogeography questions, because someone asked how to go through biogeography questions, this is how you answer it, okay? You talk about what they give you in the question and how that affects you. How that affects the species, the ancestral species, and how that eventually led to the two different subpopulations forming, two or more subpopulations forming, right? Yeah, this diagram is not that good, but okay. If you want to see, okay, look at the first row, okay? So fish A occupied the entire, between the two blue lines, that's where fish A occupied. Then after they have been separated by the barrier in the second box, after they've been separated, they form fish B and fish C. Okay? This is the correct idea of how to answer a biogeography question, or how two different fish species come about from one ancestral population. So the ancestral population here was fish A. Fish A got separated. So some fish A were at the top right corner, some fish A at the bottom left corner. And after these two subpopulations of fish A got separated, they evolved independently of each other. And one subpopulation of A formed fish B. One subpopulation of fish A evolved to fish C. That is the correct idea. Okay? Not the other two rows. If you look at the other two rows, what is wrong with the second row is that after fish B and C are separated, right, in the second, from the first to the second box, after they've been separated, fish B and fish C remain. No new species is formed. Therefore, this is not speciation. Okay? So the second row is wrong. And the third row is just completely wrong. Very, very wrong. Okay? Again, does not lead to speciation, and it cannot be that after a physical barrier forms, all fish B go to one side, all fish C go to one side. Okay, it's just impossible. Okay, so that's why rows 2 and 3 are wrong. Okay, next, someone wanted a clarification between heterozygote advantage and heterozygote protection. Okay, we go to advantage first. So advantage refers to, and this is the most common disease that we're ever going to talk about, which is your sickle cell anemia. Why it is so common? Because we can talk about it in proteins, and we talk about it in evolution and other topics as well. And gene mutation. Yes. So this part of the sickle cell content, we're only focusing on HBA, HBS alleles. Okay, so heterozygote advantage. That means that within your population, those that are heterozygous have an advantage over those who are homozygous, both for HBS and HBA. Homozygotes for HBA alleles, that means they have HBA, HBA genotype, they are selected against in malaria prevalent regions because they have a higher risk of contracting malaria. Okay, next case, your homozygotes with HBS, HBS genotype. They are selected against because they have sickle cell anemia. Okay, so now we see that if I'm homozygous for HBA, I will contract malaria. If I'm homozygous for HBS, I will die of sickle cell anemia, right? But if I'm heterozygote, I am selected for because I will not die of sickle cell anemia and I will not contract malaria, or I'll have less chance of contracting malaria, right? This is the perfect example of heterozygote advantage because the homozygotes will die, okay, very extreme, but they will die because of two different diseases. But as a heterozygote, I have less chances of dying from these two diseases. Therefore, it is an advantage, and therefore this is heterozygote advantage. Okay, so a lot of people get confused between heterozygote advantage and heterozygote protection. The main idea of advantage is you don't die. The main idea of protection is that your heterozygote individuals is that the heterozygous genotype gets propagated. Okay, so what do I mean by that? Because the diplot nature of humans allows your recessive alleles to be preserved through your heterozygous individuals, right? Because your dominant allele masks the effect of your recessive ones. Okay, so heterozygote protection means humans have two copies, right? So if I have HBA and HBS, that means if I'm a heterozygote, I have HBA and HBS. Okay, or I can have capital A, small a. That is if I'm a heterozygote. Correct? Heterozygote protection means that even though right now the heterozygotes may not be at an advantage or at a disadvantage, that means homozygotes have as much can live as much as heterozygotes can live. Okay, this doesn't make sense. Okay, let's say that again. Homozygotes and heterozygotes have the same chance of living, right? None of them are disadvantaged as of now. The idea of heterozygote protection is that they can continue to pass down both your dominant and recessive allele. That is heterozygote protection. Okay? So heterozygote protection, what it allows for, or what is the advantage of it, is because it maintains a huge pool of alleles. Okay? That means both your capital A and your small letter a, or both your HBS and HBA, right? It maintains a huge pool of alleles that might not be favored under current conditions, but could bring new benefits in the future should your environment change. Okay, so this is a thing that people always confuse themselves with. Heterozygote protection just means I have two copies, one dominant, one recessive, and I can pass it down to my future generations. That is the idea of protection. Advantage is I don't die because I'm a heterozygote. Okay. Okay, this is a question that they love. They as an SEAB. Why is the population the smallest unit that can evolve? Right? So first, whenever you tackle such questions, you need to define the definition of population, right? So it is a group of interbreeding individuals of the same species, right? Next, variation. Variation exists in a population. Natural selection acts on the individuals, yes, but results in perpetuation of favorable characteristics in successive generations. Over many generations, can you see the higher proportion of population bearing the favorable allele? Okay, since evolution is a measure of changes in allele frequency in a population over time, only a population can evolve and not individuals. So this is very confusing, right? The first time you read it, it's very confusing. But what this means is natural selection acts on the individual. This one we all know, right? So an individual organism either gets selected for or selected against. But because evolution, right, is a measure of changes in allele frequencies in a population, therefore, populations are the smallest unit that can evolve. Okay? It sounds very stupid, but I repeat, evolution is a measure of changes in allele frequency in a population over time. Okay? And natural selection acts on the individuals, but not the population. Okay? Therefore, populations are the smallest unit that can evolve. Is anyone lost? Because this is quite a stupid thing. It's quite a stupid thing. Like this explanation is quite stupid, but this is the correct explanation. It sounds very stupid the first time you hear it, but if you have a question, please raise it up. Or if you don't understand, please raise it up. Okay, nobody. Okay. So natural selection occurs through interactions between your individual and their environment, but it is the collective genetic response of the population that determines the survival of the species and the formation of new species. Okay? Okay, if no one has any questions, then I'll move on. This is not an easy concept to grasp. Okay? But if all of y'all are okay, then we move on. Okay, we move on. So adaptive radiation. This was also brought up by one of y'all. Let me explain what adaptive radiation means. Okay, it just means that there has been a rapid increase in the number of species produced from one common ancestor upon introduction to new environments. So the question that y'all raised, let me pull it up. How do we identify adaptive radiation? Okay, how you identify adaptive radiation from normal speciation events is basically, right, you look at your one common ancestor and how many species came about after this one common ancestor has been introduced into different environments, right? So if it is a rapid increase in number of species formed, it is adaptive radiation. If it is just two different species coming out from one common ancestor, it is just a normal speciation event. It is just microevolution. Okay? An example of adaptive radiation is your Darwin's finches. Okay? So we had the ancestral population of finches, but after they flew all around the Galapagos Islands, we had different species of finches afterwards. There's the most obvious example that we all know, and that is a very good example of adaptive radiation. Okay, just means that a lot of species form from one. Okay, now we'll take a short break, but in this break, you guys can try out this two questions. There's two questions here. Okay, yeah, these two. In this, 7 different types of salamanders, right? Living in mountains that form an almost complete ring around the blah blah blah. There are obvious differences between the 7 types, particularly color. Okay, so the distribution is shown in the diagram. Arrows show which types interbreed. Okay, so which statements could apply to these salamanders? First is natural selection has given rise to 7 different types of salamanders, each adapted for slightly different environmental conditions. Is this true? We see that there are 7 different types, and yeah, so therefore the first one is correct, right? Natural selection is given rise to 7 different types of salamanders. Second statement, the types are not sufficiently different from one another to be reproductively isolated. This is true as well because you see 6 and 7 can interbreed, 6 and 5 can interbreed, 5 and 6, 5 and 4, 3 and 4, 3 and 2, so on and so forth, right? So they are not sufficiently different from one another because they can still interbreed. So 2 is also true. 3, because alleles can pass from type 1 through the other types to type 7, the salamanders are named as one biological species. That's also true. And we go back to the definition of biological species is when they are not able to interbreed, right? But because they are able to interbreed, therefore 3 is also true. Therefore, the answer is A. The correct option is A because 1, 2, and 3 are all correct. Anyone have any questions on this? Okay. If not, I'll move on to the second part. Okay, this is the trickier question of the two. Okay, the coloring, toxicity, and mating calls within the populations of 170 species of frogs have gradually changed. These changes are related to the need to attract mates and the effects of predation. A study found that the more toxic the male frog, the brighter the coloring and the longer and louder it calls. Non-toxic males camouflage, call briefly from less exposed positions. Which of these statements explaining whether these changes are an example of micro or macro evolution could be correct? So first we have to identify whether this is micro or macro, and it is confusing because it is either or, right? So if you look at the options, immediately you will know that the answers are either A or C, because A means that this entire question is talking about macroevolution. Option C means that this entire question is talking about microevolution. B and D choose 2 and 3, right? And because 2 and 3 are both microevolution explanations, therefore B and D are both wrong. Is anyone confused at this point? Okay, before I even look into the proper details of it, I look at option 1, right? Option 1 is talking about macroevolution. Option 2 and 3 are talking about microevolution. Therefore, the answer is either option 1 alone or option 2 and 3 together. It can be 2 by itself or 3 by itself because 2 and 3 are both talking about the same thing, microevolution. Okay? Yes, it is hasty to come to that decision or come to that conclusion. But sometimes if you're running short of time, that's what you have to do. Okay, so the answer is either A or C. Let's look at the question in more detail. Okay, so the option 1, these changes are an example of macroevolution because they involve 170 species of frogs that show variation between the different species. At first glance, this looks correct. Okay, so it is talking about 170 different species. That's true, but we're talking about variation between different species, or we're talking about variation within each species. Okay, the latter is the correct one. We're talking about variation within the species because, okay, let's go down. Because we are talking about the changes related to the need to attract mates and the effects of predation, right? We are not talking about this between species. We are talking about within, because you see where the arrow points to, we're talking about within the populations of the species. So therefore, this is not macroevolution. Therefore, A is out of the question. Therefore, C is the answer. Okay, this is the hasty conclusion. C is the answer. Let's look into detail why C is the answer. Okay, so option 2 is correct because these changes are an example of microevolution, since they can be explained by effects of natural selection among acting only within your population. That's true because the question told you is within the population. C, this example is not macroevolution. 3, this example is not macroevolution since the only changes are in coloring, toxicity, and mating calls within population. That is true as well, right? Because there is coloring, toxicity, mating calls are, okay, maybe not mating calls, but coloring and toxicity have to do with allele frequencies, right? The different alleles will code for different colors, different how toxic they are, and then we are talking about the changes of these allele frequencies within your population. Therefore, it is microevolution. Therefore, the answer here is 2 and 3. Therefore, the answer is C. Okay. Yeah. Does anyone have any questions at this point in time? If you do, you can just say. If you don't, we'll move on. Let me check the chat. Okay. Okay, then we go through a open-ended, very fast, open-ended question. Okay, so this says, it is evidenced that pupfish once lived in a series of interconnected desert lakes. 4 species of desert pupfish can be found in the Death Valley region of Nevada, since the extensive lakes that existed there were reduced to isolated pools 20 to 30,000 years ago. Explain how the drying up of an extensive lake system to just a few isolated pools could have resulted in the development of the current diversity of desert pupfish in the Death Valley region. So the first thing you have to do is try to identify factor X. What is this factor that has led to the disruption of gene flow? Right? Always identify that first. So, does anyone want to say what the factor is? Anyone want to say what the factor is? What this factor that led to the disruption of gene flow is? No? Each species from one population. Each species from one population. What do you mean? No, the question is, what has led to the disruption of gene flow? Can you answer that question? Does anyone know? Why? What part of this question states that there has been disruption of gene flow? Just unmute yourself and say. Anyone want to say anything? Geographical isolation, exactly. Okay. Yes, it is geographical isolation. Right? So a physical barrier has come about. Okay, so this physical isolation is your is the walls or is the dry land between your different pools. Okay, because it has dried up, therefore there are spaces between these pools that are dry and your fish cannot cross. Therefore, each subpopulation within the pools have been have been isolated physically. Okay, so that is your factor X that has led to gene flow being disrupted. Okay, so first part, isolated pools mean that there is geographical isolation with a physical barrier. Next, what this means is they can no longer interbreed and gene flow is disrupted. Okay? The isolated pools present different niches or different selection pressures. Right? Individuals with favorable traits have a selective advantage and are selected for. Again, this is all very basic, right? We're just following the normal format of how a speciation answer is like. Okay. Then, of course, we talk about the change in allele frequency, so increasing frequency of favorable alleles. Different subpopulations evolve independently of each other. Their allele frequencies changed, accumulate different genetic mutations, subjected to genetic drift and natural selection over a long period of time or over hundreds and thousands of generations. This led to reproductive isolation and the formation of 4 different species. Okay, if you want to stick very obediently to the format, okay, what is left out in this explanation is also the mentioning of the common ancestor. Yeah, that's not mentioned in this. And then we didn't talk about the production of viable fertile offspring. Okay, but yeah, this is also an accepted answer. It is, you just find whatever helps you. Okay, if you feel like a format will help you, stick to the format that we talked about above. We'll share the slides so y'all will have that format. Yeah, stick to whatever format you are comfortable with. You can use mine, you can use your own, as long as you have a format that covers all that you have to cover. When you see an exam question on speciation, you will start laughing. Okay, behind your mask, you will start laughing because you'll be like, haha, I have the perfect answer. I'm going to get all the marks here. If it's I don't know, if it's an essay question, let's hope it's an essay question. If it's an essay question, you'll all have that, 8 marks secured. Okay, you'll see the question. When you're like checking the pages, right? Teacher asks you check the pages. You'll see this question. You'll be laughing behind your mask because you'll already know what you're gonna answer, what exactly you are gonna write, because you have your format.